This video will show you how to explore one residue in a protein structure. The protein structure we'll use is human pancreatic alpha amylase. So we'll search what structures there are here. It's related to the one that we used in the lab on Monday, but I thought it would be interesting to look at the human one. So there's 40 structures it found, and we'll take one that's interesting because it's in complex with the potential drug. So it's in complex with myricetin, and it turns out the human amylase is an anti-diabetic target. So we could explore this further here. I'll click it and open it, but really what we know now is how to access that data set. So we need to know it's 4GQR, that's the structure. But here's the publication details, and we can also check out the sequence if we want to know which residues have which secondary structure. The residue we'll look at today, and this is kind of random, is residue 303, if I'm not mistaken, 336, sorry. So that's down here, it's a threonine, and it's part of a beta strand. Okay, so how do we look at the structure? We go to Firstglance in Jmol. If you Google this will come up. I already put in the right PDB identifier, and then we can submit that, and it will show us the structure. This will take a while to load, especially if it's the first time you use the program, because it has to download some applets. Okay, here's the protein structure. As I said in class, it rotates. If you want to turn that off, click on spin here to uncheck that button. And then we can take a look at it in terms of secondary structure. And what we discussed in class is that we have a couple of beta sheets that are sandwiched together, so they're right on top of each other. We also have this beta barrel in the center, and we have various alpha helices around that central beta barrel. The drug that's bound to this human enzyme is over here, and the green spheres, there are calcium ions. So this one might be a chlorine, and this is a calcium. So this is also a calcium dependent enzyme. And you can imagine that the active site is close to where the inhibitor binds. Okay, but we want to zoom in and look at that one residue, 336. So let's find that residue. Just type in 336, and it should highlight. If we want to see all the highlights easily, we can click on here, and it just shows the backbone. Okay, and undo that. So the residue we're interested in is here, and you get the sense that it's in between. Let's zoom in a bit. So to zoom, you can take the middle mouse button if you're on a Windows machine, and so it's coming off of a strand, and it is close to side chains from two alpha helices. So that's the general area. It's pretty crowded right now, so we'll use the contacts command to just show the residues that are close. And the way we do this is we click on atom with halos and then show atoms contacting target. And it's still pretty busy, so we can turn off the background, we can center the contacts, and we can use this view where they use ball and stick for our residue of interest, and these stick drawings for the rest of the contacting residues. Okay, and if this is still too complicated, we can also turn on this lab, and we can turn off the halos, so it's not so busy. Okay. So all the atoms with a star are the atoms in threonine. If we want to verify this, we can click on the atoms. So this is the nitrogen, this is the C alpha, this is the carbon of the carbonyl, and this is the oxygen of the carbonyl. So that's the main chain, and on top here is the side chain. Off of the C alpha we have the C beta, the methyl group on the C gamma, and then an OH group, which is called O gamma. We don't see the hydrogens because they're not resolved in the crystal structure. Okay, and so now we can see what's close to these atoms. So threonine has a hydrophobic part, that's the methyl group, and it has a hydrophilic group, that's the hydroxyl group, and you can already get the sense that there's lots of other oxygens close to this oxygen, and that there's lots of other carbons on this side. So, for instance, here we have a valine that's close to the methyl group. You can see how close. So this is a good non-bonding distance, more than 3.5 angstroms. So they're in contact, but they're not clashing. And up here is another hydrophobic side chain. This is the methionine. Just trying to get the distance here. That's 3.62 angstroms away. They're giving you 2 decimal points, but you can basically forget about the hundredth of an angstrom unless you have very highly resolved structures. Okay, let's look at the other part of the side chain. So now we're talking about this OH group of the threonine, and we can see what's close to that. If you're not sure which atoms are close, you can just double click on here. Let's try that again. There we go. And just touch the atoms. So this is 2.6 angstroms away, this is 3.2 angstrom away, this is 3.35 angstrom away. And typically for distances lower than 3.5 angstroms, you would expect that there's a hydrogen bond going on or some other interaction. So this is the closest one, so that's the one that we'll mark up. It's part of the side chain of a serine, so the OH group of the serine and the OH group of the threonine come close together. Okay, so let's look at the main chain interactions. We say the main chain is hydrophilic because it has hydrogen bond donors and acceptors. Here's the carbonyl group, and this carbonyl oxygen is closest to a nitrogen on a different chain, so it makes a main chain hydrogen bond. If we turn on the secondary structure, we'll see that those are 2 strands, and this is the typical hydrogen bonding pattern. So here we have the nitrogen, and that's a bit too far for a hydrogen bond, so I won't show this, but instead it's close to this water. So this is an unusual hydrogen bonding pattern for the nitrogen. It's not really on the surface of the protein, but it has the water close by. Okay, that's almost all we want to do. The last thing I want to do is turn on the secondary structure, and that's a bit involved. You have to type something. So first you right click on here to get the console, and then you can type commands in here, and I have a cheat sheet that I'll give you later as well. So we have to say select all, and then rockets on. Rockets are the way I want to show the secondary structure, and then so that they don't block everything, we'll color them in a transparent way. It's called translucent here, in yellow, so they won't bother us too much. And then we can select none, so that it doesn't make those halos around it. So we'll run this, or you can press return, doesn't matter. And then we'll get this window back, and so now we can see the secondary structure. got a bit busy, so maybe I'll turn the slab even lower. But now we have a nice view of what's going on here. So this threonine makes main chain hydrogen bonds, and it's from one beta strand to the next beta strand, and the methyl group of the side chain comes close to other hydrophobic side chains, and the OH group of the side chain comes close to this serine making a hydrogen bond. The last thing we want to do is take a screenshot. So that depends on your computer, but in this case there's a button on the keyboard for it, so I'll just use that and print screen, and then I can paste that into another file. Or I can upload it on Piazza, which is what I'm asking you to do. Okay, so your goal is to pick a different amino acids in the same protein and explore the side chains interactions a bit and see in which secondary structure it is placed, and then take a snapshot and paste that onto Piazza so we can see which residue you looked at. The last thing maybe to do is click on here so we see which residue it is, and then I can take my screenshot. Okay, that's it.